I'm happy to announce the talk by Konstantin Popadin. Unfortunately, he's not here, but you've seen lots of his students who were extremely active. They had that special corner over there, and nobody came alive out of that corner. I've observed it. So Kostya decided to stay away from all that, all these events. Kostya, welcome. And the talk will be about I'm sorry, I don't see it. A to G substitutions as a hallmark of oxidative damage in mitochondrial and bacterial genomes. Kostya, welcome.
Yeah, thank you very much. So I hope you can hear me and you can see my screen.
Fine.
Good. Okay, so today I will present this story about one mutation signature, which is favorite mutation signature in my laboratory. So, and I will go through overview of several projects and will present a strategy for, say, next 1 or 2 years, what we're going to do. Okay, let's go. First, what is a mutation spectrum? So there are 4 types of nucleotides, A, T, G, C, and there are 12 potential substitutions between them. So some of them we call transitions if they are occurring between purines or pyrimidines, and others, they're transversions. So for example, if we count number of transitions and transversions, it's already kind of mutation spectrum. So this is some characteristic of mutation process. If we want to go deeper, we just can count 12 types of substitutions from G to C, from C to A, etc. So in this case, we can call mutation spectrum as a 12-component mutation spectrum. We can go even deeper if we have a lot of data. We can take into account neighbor nucleotides. For example, 4 nucleotides before, 4 after. So we just multiply 4 by 12 by 4, and we can have 192 component mutation spectrum. So rich enough with a lot of information. So that's what we analyze for different species. And I would like to emphasize that by our definition, at least, mutation spectrum is a relative rate of different types of nucleotide substitutions which we observe at neutral sites in given gene or genome. So neutral is very important because we would like to characterize mutation process which is separate from selection. And in all my slides, I will present data which are based on analysis of polymorphisms, of synonymous 4-fold degenerative sites. So the most neutral positions which we can find in mitochondrial genome at least. Okay, so second question, why it's interesting to investigate mutation spectrum. So let's discuss this example, for example. So it was published that frequency of guanine in mitochondrial genome positively correlates with maximal longevity in different mammalian species. So here 1 dot is 1 mammalian species, and we can see that long-lived mammals here, they have excess of G, excess of guanine in the mitochondrial genome, while short-lived species, they have a deficit of guanine. And this correlation is interesting per se, but we don't know if it's a result of selection. So it means that it's beneficial to be G-rich mitochondrial genome in case of long-lived mammals. So it's like selection explanation, or it's just mutational bias. So due to some reasons, mitochondrial genome in long-lived species, there is a bias towards G. So without this deep understanding, we cannot distinguish 2 processes, and we cannot understand deeper molecular evolution. So we just can describe as it was described here. Okay, so if we want to go deep, if we want to distinguish mutagenesis and selection, we need to know mutation spectrum. And another reason to investigate mutation spectrum that if we know like frequency of different substitutions. So here I just provide piece of a mutational signature from Cosmic database, the biggest cancer database. And this is signature, it's called signature number 1 because it's the most common substitution from C to T. And we can see different contexts. So for example, if after C there is G, here, 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 and here, we see higher frequency of C to T substitutions. And we can observe, we can just read in this cosmic database that exactly this signature number one is some process which is characterized as a spontaneous or enzymatic deamination of methyl cytosine to thymine, and it behaves as if clock-like process. So, for example, it correlates with number of stem cell divisions in different tissues. So it means that if we have empirical observation of mutation spectrum, we can guess what kind of mutagen is responsible for this mutational signature. So, and it's good to know mutagens. Okay, now let's come to mitochondria. What can be, like from bird's eye point of view, what can be interesting in mitochondrial genome? So everybody knows that mitochondria is a source of reactive oxygen species, and it's a very important organelle for aerobic metabolism. So if we have something relevant in mitochondrial genome which correlates with life history traits or with basal metabolic rate of different species or with oxygen consumption of different species, we expect that this process can shape rules of mutagenesis of mitochondrial genome. So that's naive hypothesis. And here I would like to emphasize that in case of mitochondria, there is a asynchronous replication. So one big part of mitochondrial genome is during replication, it happens in single-stranded state for a long period of time. And exactly this single-stranded mode of DNA is it's prone for mutations. So that's why majority of data which we obtain in our laboratory, they are coming from genes which are located on this strand, which is called heavy chain. And with this heavy chain is single-stranded for a long period of time. So many mutations which we observe, they occur in this single-strand condition. So SS marks single-strand DNA. So because mitochondria is involved to determine, to shape level of aerobic metabolism, we hypothesize that mitochondrial mutation spectrum somehow can correlate with the life history traits of different organisms, because life history traits of different organisms correlate with level of the aerobic metabolism through reactive oxygen species. This is main hypothesis. Okay, as soon as we have this hypothesis, we go to literature and we try to find what is the best known mutational signature of reactive oxygen species. So what do we know from cancer research, etc. And we find this transversion G to T. So, and indeed, there are several molecular signatures in cosmic database, signature number 18 and 36, which are well proven in the nuclear genome that and it was even proven experimentally in when cells were exposed to free radical species. Okay, so we test if in mitochondrial genome we can observe G2T mutational signature, which should correlate with aerobic level of metabolism. And here, let's have a look on average mitochondrial mutation spectrum. This is just average from all mammalian species. And if we find G2T, it's here. So it's less than 3%. So for example, T2T is more than 60%. It's very common, it's the most common transition. Second most common is A to G, about 20%. But G2T, which is expected to be high in mitochondria because mitochondria is associated with ROS, and because G2T is a hallmark of ROS, it's just 3%. And so here I provide our results from mammalian data, but if we go to literature, we read cancer papers, papers from somatic mutations, the results are the same. So nobody can observe significant fraction of G2T in mitochondrial genome. And that's why this problem is open. And we don't know why in mitochondrial genome, nobody observes expected signature of reactive oxygen species. Okay, you can say, even if this contribution is very low, like 3%, but maybe it will have some effect when we compare different species, for example. Okay, so we decided to test it, and we reconstructed mutation spectrum for hundreds of mammalian species and performed principal component analysis. So here you can see by plot, just projection of different substitutions on principal component 1 and PC2. And according to our results, PC2 perfectly correlates with generation length of mammals. So long-lived mammals would be here, short-lived mammals would be here. And we can see the GTT, so it's here again, so it's not significantly affect principal component 2. So even, so two problems. The absolute ratio is very low. And the second, this weak effect doesn't demonstrate any correlations, any biologically meaningful correlations with expected life history traits, with generation length of mammals. So we can ask why we don't see this effect. So maybe mitochondria just well protected against reactive oxygen species. Should be. That's why maybe we just don't have this effect at all. Or maybe in mitochondria there are some specific types of reactive oxygen species, because reactive oxygen species is a complex family of short-lived chemical mutagens, and that's not so simple to investigate it. Okay, so we don't know. We don't know answer, but we decided, okay, let's go for data-driven approach, and we will compare many species, and we will see which category of mutation can discriminate species with different life history traits. Okay, and that's what we did. Let's start from the middle. So first of all, we just compared hundreds of mammalian species. For each mammalian species, we reconstructed mitochondrial mutation spectrum based on polymorphic data from GeneBank. And we observed that short-lived mammals, they demonstrate low fraction of A to G substitution, and long-lived mammals, like elephant, they demonstrate high fraction of A to G. And A to G is the only type of substitutions out of 12. So we checked all of them, which demonstrate significant correlations with life history traits. And in this case, life history trait was generation length in mammals, because really they correlate with each other. So basal metabolic rate, longevity, number of offspring, etc. They perfectly correlate with each other. Okay, having this result, we decided to extend this logic, and we asked it, okay, because mitochondria is transmitted through oocytes, and mouse oocyte and elephant oocyte differ in age, let's say how many years oocytes spend in post-mitotic state, maybe we can see the same effect if we compare population data from human. And so we compared de novo mitochondrial mutations from different families, from families with different woman fertilization age. And we observed that mothers who are age matched, their offspring have excess of A to G substitution as compared to offspring of young mothers, so which is completely in line with these mammalian results. Okay, so and after we decided to extend logic even more, and we decided, okay, let's go to different cells, because cells can be short-lived, like colon epithelium, and they can be long-lived, like skeletal muscle or neurons. And if mutation spectrum, which we observe on the level of different species, different organisms, population, is universal, maybe we will see the same pattern. And we analyzed the biggest cancer data set, and we would confirm it that probability of A to G substitution is increasing in long-lived tissues, like neurons. Okay, so the explanation for all these three pieces of result was that with aging, with cellular aging here, or with oocyte aging here and here, there is like oxidative damage. And A to G is a substitution which is sensitive to this oxidative damage. Okay. Next, we decided, but if we take some species with similar generation lengths, so we will exclude this aging factor from our analysis. What we can see in this case? Okay, so in order to do it, we controlled for generation lengths. Let's say we compared different ectotherm fishes with similar generation lengths, but different temperature. And we observed that the same mutation A to G positively correlates with temperature. And of course, temperature in case of ectotherms, in case of fish, is a just very strong approximator of basal metabolic rate or oxygen consumption. So oxygen consumption is expected to be very strong in tropical fish as compared to north cold water fishes. So we can say that the same signature is associated not only with aerobic metabolism, with aerobic damage, which is associated with aging here, but it's also associated with normal basal metabolic rate, which is driven by temperature in this case. Okay, next we extend all our analysis, and we have first interesting pilot results comparing insects. So we compare short-lived cockroaches with long-lived social insects, termites, and we see the same situation which we observed in mammals. So short-lived insects and long-lived, and in termites we have excess of A to G substitution in mitochondrial genome. And very recently, so I think this poster was presented also on the conference, we observed that mitochondrial mutations A to G, they have excess in birds with outstanding flying capacities as compared to chondrials. And again, we expect that birds with species with outstanding flying capacity most likely have increased basal metabolic rate and as a result, increased oxidative damage in mitochondrial genome. Okay, so that's what is happening now in our laboratory, and we try to expand logic every day. And for now, the like intermediate conclusion that A to G is a signature of oxidative damage in single-stranded mitochondrial DNA. So single-stranded because majority of protein coding genes, which we analyze, they are located in this heavy chain, which I introduced in the beginning. And luckily, we found 3 experimental papers which prove that A to G can be really sensor to oxidative damage in single-stranded piece of DNA. And these papers, they confirmed it for in case of Escherichia coli, yeasts, and aged mouse oocytes. So we believe that this is a more or less universal result. And if this is universal, we may try to expand it to bacteria and to prokaryotes in general. And so next, our question, if prokaryotes demonstrate a ROS signature, and if they demonstrate something like this, is it G to T, which is classically known and well accepted, or it's A to G, which is described for now only in mitochondrial genome. And so why we decided to test prokaryotes? Because mitochondria is descendant from prokaryotes, from bacteria. And we hope that mutational rules can be similar. And so we started just to investigate literature, and I would like to remind you this evolutionary paradox. Briefly, I will walk you over classical papers. First of all, GC content in prokaryotes is characterized by huge variation from 25 till 75%.
Kostya, please, not all classical papers.
Not all.
You have minus 1 minute left.
Yes, yeah, that's my last slide. Yeah, I'm sorry. I'm catching up. So this GC variation can be explained either by neutral mutation bias or by selection. And still now, nobody was able to explain it by selection with some universal mechanism. So most likely, this variation is significantly driven by mutational bias, so it's neutral. And now the most interesting paper is Naya et al. The figure is here, that if we compare bacteria and archaea with different species, so aerobic and anaerobic, we do observe different GC content. For example, in the firmicutes, in protobacteria, and euarchaeota, always GC content in anaerobes is less as compared to aerobes. So now, if we want to explain this result as a result of classical G to T signature, Well, we cannot do it because we expect opposite. If aerobes, they have a lot of G to T substitution, they should become G poor, but in reality, they are G rich. So our hope, our working hypothesis that instead of G to T, we need to look for the same mitochondria specific substitution A to G. So A to G is like a hallmark of oxidative damage. And if we use this A to G substitution, this paradox should be solved. And for now, our pilot analysis, they in line with this paper, but now we just derive species-specific spectrum for each bacterial species in order to prove it finally. And so my take-home message that I would happy to add to this general overview, this aerobic prokaryotes versus anaerobic. And so that's our working hypothesis till now. And I would like to say thank you to my hardworking and very effective laboratory. A couple of relevant bioarchives are here, and you're welcome to our lab webpage and lab YouTube channel with more details. Thank you very much.
Thank you.